All right. Okay. It's a bit strange because I'm alone in my office, but I need to give a presentation. But anyway, thank you, Dr. Yoon, and thank you, organizer, for the World Innovation Forum. And I am really excited to introduce our kind of a research, and I'm honored to be a speaker for this wonderful World Innovation Forum. So today I'm going to introduce the one of direction in our lab to explore the develop the human brain region, especially the area called the diencephalon. And we try to reproduce the developing human diencephalon region by using the human brain organoid. So basically, our lab overall interested in studying the human brain development, and also we are interested in using human brain organoid to development, but also the study the human brain disorders. One of the simplest question for us is that how we really reproduce this the human brain development and maybe disorders in vitro. So over the years, people have been studying the brain development, not just the human, but animal, including the mouse, zebrafish, and even Drosophila, to understand the fundamental biology of the brain development process. Especially in the early embryonic stage, there arise the primary vesicles from the urulation at the urulation stage. Especially there are like telencephalon, diencephalon, and mesencephalon, metencephalon, and myencephalon that represent the different parts of the brain in adult. Uh, for today's talk, I'm going to focus two areas, telencephalon and diencephalon. Especially telencephalon quickly introduced to produce the cerebral part of the brain. But mainly today, I'm going to introduce the brain area developed from the diencephalon, thalamus and epithalamus. So basically, there are many methods to produce the brain organoid, but we are using so-called like region-specific brain organoid regeneration by using guided approach. So in later, Dr. Chang Pak will introduce the morphogen gradient to introduce the different neural axis of the brain development. But today I'm going to kind of just introduce the dorsal ventral part of the brain development. In few years ago, we uh, established and published the method to produce dorsal part of the human brain. We simply name them as a human cortical organoid. Basically, this human cortical organoid represent dorsal cortical area of the human brain. We also used a ventralizing morphogen called sonic hedgehog to ventralize cortical organoid to produce the ventral cortical organoid. Simply, we named them as human medial ganglionic eminence organoid. These organoids form the ventricular zone, like shown here, but also, as you can see here, they make kind of a not complete, but the extensive enough the laminar layers of cortex. To see the function of this telencephalon, we knew that they are directly connect to the subcortical area called the thalamus. So we made an effort to produce the thalamic areas in the brain by using the human stem cell. So basically, we introduced BMP7 and some kind of FGF signal inhibitors to produce this thalamic organoid. And as you can see here, this thalamic organoid express thalamic marker, pan-thalamic marker called the T7L2, but they don't express the cortical marker TBL1. So as Dr. Yoon introduced, by using this region-specific brain organoid, we showed that the assembly or fusion of cortical organoid with the ventral cortical organoid can reproduce migration of GABAergic neuron from ventral to dorsal cortical area. But by assembling this thalamic organoid with the cortical organoid, we also showed that this assembloid can reproduce corticothalamic thalamocortical projection. So while we are working on this kind of assembloid, we thought that maybe this different part of the brain area, kind of we could further define really the small area of the brain region. For example, although I introduced cortical organoid, MG organoid, and thalamic organoid, if you look at the human brain and animal brain as well, this cortex is really complex. It has a prefrontal and visual cortex as well as somatosensory cortex. But even in the thalamus, although the thalamus is much smaller than the cortex, thalamus has a lot of the nuclei that connect to the different parts of the somatic area, peripheral area, but also different parts of the cortex. So for example, there are anterior nucleus to the kind of a medial lateral nucleus in the thalamus that connect to the different areas. So in our lab, one of the main effort is to reproduce the different area in the thalamus and cortex by using the brain organoid. So this is our goal. So because not many people are familiar with the thalamus and the diencephalon development, let me quickly introduce the development of the diencephalon. So basically, diencephalon developed as the unit called like prosomere, simply named it as a prosomere 1, prosomere 2, prosomere 3. P1 that represent the pre-tectum area, and P3 represent the pre-thalamic area, and P2 mainly represent the thalamus-related areas. As you can see here, because the thalamus, we have this rostral and caudal thalamus, and those are the kind of a term people use. But because the location, this rostral thalamus is also called the ventral thalamus, and the caudal thalamus is called dorsal thalamus as well. Then how can this develop different parts of the diencephalon develop? As I introduced already, this developmental cell fate is regulated by the morphogen gradient. A very simple rule is that the sonic hedgehog induced the ventral fade throughout the neural tube, including telencephalon, diencephalon as a middle and hindbrain. And the FGF signal as well as BMP4 represent induced dorsal fade. So because we already had the method to produce the thalamus, we had a very simple idea. So maybe by adding the sonic hedgehog or BMP and FGF into the thalamic organoid, we may be able to either ventralize or dorsalize the developing thalamus. So those are exactly what we tried. Basically, we added a ventral fade inducing sonic hedgehog during patterning step of the thalamic organoid production. Then how we can kind of identify either dorsal and ventral fade of the thalamus. So previously, development biologists already defined a gene that mark the dorsal and ventral fade of the thalamus. So for example, LHX2 shown to express highly in the dorsal faded thalamic area, and LHX5 expressed in the ventral fade of the thalamus. So by using these two protein expression, we're trying to define either dorsal or ventral fade of the thalamus. Without adding any sonic hedgehog, so when we stain the thalamic organoid with the LHX2 and LHX5, as you can see here, this thalamic organoid highly express LHX2, meaning that they're mostly the dorsally faded, and there are small number of cells that express the green LHX5, but number is quite small. However, when we added sonic hedgehog, so now you see that this ventrally faded thalamic organoid contain the cell that express LHX5, which represent that they are really represent the ventral part of the thalamus. Moreover, we try to understand what are the neural fate of this dorsal and ventral faded thalamic organoid. So very simply, we used two marker, one marker for the excitatory neuron, the VGLU2, and GABA as an inhibitory neuronal marker. As you can see here, thalamic organoid mainly express the VGLU2 representing they represent excitatory neuron, and this ventral thalamic organoid express GABA, meaning that they are mainly the GABAergic inhibitory neuron. So furthermore, we were interested in how this dorsally faded thalamic and ventrally faded ventral thalamic organoid show the neural activity. Here we used Voltron to measure neural activity. So basically, in these two type of organoid, we used the AAV virus that express Voltron, and we measure neural activity. So as you can see here, both thalamic and ventral thalamic organoid show neural activity, but especially the ventrally faded thalamic organoid show bursting type of neural activity, which is known to present in the specialized thalamic nuclei called TRN, thalamic reticular nucleus. So right now, we are trying to assemble this thalamic organoid with the ventral thalamic organoid to understand the interaction of excitatory neuron in the thalamus and the GABAergic neuron in the TRN and the ventral thalamic organoid. Especially this connection is really important for the epilepsy as well as some other kind of neuropsychiatric disorder. So we are trying to model those diseases by using the model. Then how about the dorsalization of the thalamic organoid? So as I introduced, sonic hedgehog-induced ventralization of the thalamus, but BMP4 and FGF8 is known to induce dorsalization. So again, very simply, during the patterning step of thalamic organoid development, we added FGF8 and BMP4. And we produced the organoid, and we just simply named them as dorsal thalamic organoid, assuming that this BMP4 and FGF8 induced dorsalization. Here, very interestingly, I haven't introduced, but in the dorsal part of the thalamus, there are two main areas. They're named as habenula and pineal gland. We expected either pineal gland or habenula could be produced from the BMP4 and FGF8 induced thalamic organoid, but we found that only the pineal gland marker, CRX, LHX4, and BSX are expressed in the dorsal thalamic organoid. And these are the qPCR and immunostaining data. And also we found that the melatonin synthesizing enzymes are expressed in the dorsal thalamic organoid. And of course, when we look at the expression of melatonin, so this dorsal thalamic organoid express melatonin. So right now we rename this the dorsal thalamic organoid as a pineal gland organoid, and we're looking at the function of this pineal gland organoid in vitro and in vivo by transplanting the pinealocyte as well as the pineal gland organoid into the animal brain. So in conclusion, even in the diencephalon, we could induce the ventral fade by using sonic hedgehog and the dorsal fade with FGF8 and BMP4. And the ventrally faded thalamic organoid produce GABA neuron, and dorsally faded diencephalon behave like a pineal gland, and we named them the pineal gland organoid. And they produce melatonin. All right. So I want to thanks to Ridvan who performed all the work that I present today, the ventralization and dorsalization of the thalamic organoid, and we have other investigators in our lab working on different kind of a project. All right, thank you for your attention.
Okay, thank you very much for a wonderful presentation. And we actually having some time to get questions
Oh,
from the audience. Uh, so you can ask directly to turn on your audio, or you can also write your question on your chatting board on side.
right, sure.
Uh, can I ask a question?
Sure, yeah, Professor Johan.
Thank you, Professor Bach. I have actually very simple questions. So in your diverse type, different type of the organoids, can you obtain the astrocyte if you continue to differentiate them?
Sure. So we haven't tried to isolate astrocyte, but according to our single cell analysis, over 20% cells show the astrocyte marker. So definitely there is a way to kind of dissociate and expand. We only try to dissociate and expand the neuronal cells to measure neural activity and performing some other experiment like proteomics, but we never tried astrocyte isolation, but definitely should be possible.
Yeah, I think that if you can dissociate the astrocyte from the different organoids, maybe you can classify the subtype of astrocytes in the different area of the brain region. So that will be really great. Thank you.
Sure, great idea. Yeah.
Okay, any other questions? While waiting the questions from audience, I have one quick questions. I wonder, I mean, you are developing many new regionally specified organoid system, and I wonder how you define the optimal concentration of morphogens such as FGF8 and BMP4, sonic hedgehog. I mean, sometimes just using sonic hedgehog to ventralize the organoid, it just lead to the median ganglionic eminence fate instead of the thalamus. I don't know, as also protocol developer in organoid field, I really wonder what is the easiest way or the efficient way to develop the concentration optimization in organoid system.
Right. So I think there is no easy way unless we maybe have some kind of a machine that you can kind of screen the multiple conditions. But in our case, we use different concentration of sonic hedgehog as well as different concentration of ARC and a BMP7 kind of 7. And we also tried a different duration of treatment. So although we only, I mean, here I only mentioned that day 7 to 14, for example, we also tried like a 10 to 18 and then maybe the 14 to 28. And there are a lot of kind of condition and optimization we tried, but I haven't present. But so basically, we have to kind of try different conditions.
All right.
Right.
Thank you very much.
Sure.
Any other questions from the audience? I just got one more tiny questions. So you developed really nice system of ventral and dorsal thalamus. So are you thinking about to investigate certain specific disorders of brain using this new model system?
Right. So we are thinking about epilepsy. One of our graduate students, she's very interested in epilepsy, and we know that. I mean, people already know that the thalamus, together with the TRN kind of interaction, is critical for the absent epilepsy. So we're trying to we are right now in the process of fusing them, making a symboloid, and measuring the neural activity and to induce the epilepsy and see whether we see some kind of epileptic kind of neural activity. And also, we have a few disease models. I mean, the iPSC from the disease, such as Angelman syndrome, as well as Rett syndrome, as well as a few autism spectrum disorder. And some of those disease shows some epilepsy. So we're trying to see whether we can model those. So those are the one area that we are thinking about. I mean, we are doing right now. Yeah.
Yeah, that sounds very exciting. Okay, so it's time's up. So thank you very much